The endocrine system. Anytime we talk about a system, we're really talking about a collection of organs. And really what a collection of organs is a collection of cells or tissues, and tissues are just a collection of cells. So when we look at a system, we're looking at basically groups of cells that interact. And so what we're looking at is the endocrine system. What does endo mean? Endo means in, and crine has to do with a signal. So what we're looking at is an internal signaling system. And the reason that we need an internal signaling system is to respond to varying internal and external changes. So we're going to see endocrine systems in organisms that are multicellular, in organisms that have many cells that need to be able to coordinate their response to some sort of change, either an external change or an internal change. So this really falls under the category of regulation, or being able to control. Another word that comes up very often is homeostasis, which is the ability to regulate or to try to maintain a consistent level. And the way that we do that is with a signal. This signal needs to be released. This signal needs to be received. And the signal needs to have an effect in order for it to truly be an effective signaling system. And so the response is really the effect. The signal doesn't mean much unless something actually happens. The main way that organisms, plants, animals, fungi are going to communicate is with hormones. Hormones can come in many different types. We're going to see the main types being protein or lipid hormones. And the main idea is that hormones are signaling molecules. Now, it's not the only way that you can send a signal. The animal body especially will also use electrical signals through the nerves to be able to send nerve signals, as well as to coordinate the contraction of the heart muscles, which we'll look at later. But when we look at these signals, we actually kind of have three different types of signaling effects. Paracrine is going to be local signaling. And this means there is no wide transport. So this means that whatever signaling molecule we're using in a paracrine signal is not going into the bloodstream. It's having an effect in the immediate local area. Endo and exocrine, these are going to travel to their target, usually in the bloodstream. Now, plants will also use hormones. They don't have a bloodstream, but they do have a system of tubes that can transport through their bodies. Endo and exo, you don't necessarily need to know the big difference between them. Endocrine do not require a duct, and exocrine do require a duct. Basically, it's just how is it released from the gland itself, and we don't really need to worry about that as much as, is it a local signal, is it a long-distance signal, and pheromone is actually an external signal. Pheromones have long been associated mainly with insect communication. We'll look at this later when we look at some innate behaviors of animals. The ability to communicate is innate. And so insects have long been studied to use this. This is how ants create a trail. This is how honeybees communicate.
Darius Reed, please come to the front office. Darius Reed.
It does turn out that other animals, including humans, actually use pheromones. They believe that pheromones were once being featured in a movie called Love Potion #9, because they believe that pheromones are part of what makes you attracted to someone else. So what we need to do today is we need to talk about what is the structure of an endocrine system. This is what it does, but now we want to look at how does it do that. So when we look at the structure of an endocrine system,
Colonel Laughlin, please call the front office. Colonel Laughlin, please call the front office.
we're going to have glands, and these are groups of cells that create and secrete hormones. And if you look at the top picture, you actually see a gland on top of the kidneys called the adrenal gland. You actually see the thyroid gland as well. And so it's just a collection of cells that's going to create and secrete a hormone, but usually in response to another signal. So interesting, which signal comes first? When we look at the types of hormones, this is an important category to make sure that you pay attention to, because the type of hormone is going to have a big effect on what it can do. Polypeptide, this is really another word for protein. And so going back to thinking about many peptide bonds, and so what we're going to see is just differences in, does it have, it definitely has primary protein structure, which is the amino acid sequence. And then we're going to see the secondary, tertiary, or possibly quaternary structures as well. But the key about a protein is that it's pretty large. However, it's able to be very specific. If we remember back to those protein folding, a protein is a very, very specific shape. So it's able to be unique in its shape and be very effective at activating only its target. Because the target is the cell where we want to produce an effect. And keep in mind, cells are differentiated. Not all cells can make insulin. Not all cells should make insulin. It's the idea of differentiation within the animal body. And so we do have to really pay attention to this is an advantage, being able to target only certain cells. And so proteins are very specific in their shape. Next we have glycoproteins. Glycoproteins are going to be very similar to polypeptide, but they do have an added carbohydrate, which can help with binding to the target. Both of these are actually going to have to bind to the target, and usually amines are as well. Amines are derived from amino acids. So if you remember your basic amino acid structure with the central carbon, the other carbon with the amine group, and then the other carbon with the carboxylic group, it's still a pretty big molecule, and it's still usually polar. Steroids are going to be lipid-based, and the key thing here is that they are non-polar. Why is this important? We're trying to send a signal to a cell. We want the cell to do something inside of the cell. We don't just want the signal to get to the cell. We want something to happen on the inside. If I'm using a non-polar signal, it can diffuse across the cell membrane. Now, it is going to need a delivery vehicle to help it transport through the bloodstream, but once it arrives at the target cell, it can diffuse across the membrane. Polar signals, your polypeptides, glycoproteins, and amines, are going to need to bind to a receptor. And that receptor is what's going to have an effect on the inside. So once the signal reaches its target cell, it will do one of two things. If it's lipophilic, if it likes lipids, it is going to diffuse across the membrane. The advantage of this is that molecule can have a direct effect on the cell. It can activate a molecule on the inside. It can bind to another molecule and turn it off. It can have a direct effect. If we're looking at a lipophobic, which is a hydrophilic, which is a polar, going back to your biochem, lipophobic are not going to be able to cross the phospholipid bilayer. Lipophobic must bind to a receptor. Now they can have an action, so we're going to see ligand-gated channels when we look at the nerve cells and something binds and the channel opens. So they can either have action externally, or they can activate an internal molecule. And the most common internal molecules to see are what are called second messengers. So the endocrine system has produced a hormone that has arrived at the cell that has bound to a receptor or diffused across the membrane and has then had an effect inside of the cell. The most common effect is actually either going to be activation of an enzyme or gene regulation. And so I really liked this diagram because it shows you the G protein receptor. So the hormone binds to the G protein receptor. The G protein actually activates another protein. Here it's an enzyme called adenyl cyclase, which turns ATP into cyclic AMP. CAMP actually is able to move across the nucleus and have an effect on a transcription factor, which will allow transcription to either occur or to limit transcription. So the key here is, what is the hormone and what is its possible action? The next page of the notes, I just want to kind of look at what is feedback. How do hormones regulate body systems? The two main ways is by either positive feedback, which means the increase of a signal increases the effect, or negative feedback, which is the increase of a signal inhibits an effect. The main example with positive feedback we're going to see is something called oxytocin. Oxytocin is a hormone that causes uterine contractions or childbirth. But there's also a hormone in fruits called ethylene that's going to stimulate ripening. What this means is the more I have of it, the faster that process happens. And that is one huge advantage, to be able to create an amplification effect of the more I have, the more it happens. However, a lot of times it's actually better to control things by turning them off, allowing them to function normally until I need to turn them off. So really, if you think about positive feedback, it's probably normally off and then gets turned on, similar to our promoters and our operons in gene regulation. In negative feedback, we're actually going to normally be turned on, and I'm going to increase a signal to inhibit that effect. So if you look at this little flow chart, the hypothalamus creates something that turns on the pituitary, that turns on the target gland, that creates the hormone. Large amounts of this hormone, if I have enough of it, are actually going to go backwards and negatively feed back on these two mechanisms. One of the big things that we want to see is the idea of conformational changes. A molecule being able to bind and have a direct effect.